So, hi everyone. Uh, Welcome back to today's session. I am Aishika Goswami, and before diving deep into the topic, let me just give a brief introduction about myself. I have qualified 26th West Bengal SET or State Eligibility Test. Along with that, I have qualified CSIR NET JRF December 2024 with an All India rank of 61. I have also qualified GRADE Excel 2025 with an All India rank of 85. And this year, I have also qualified the BARC OCES examination as well as the interview for it. I have also been selected for PhD at IISc Bangalore in both biochemistry as well as in molecular biophysics unit. So if you are someone who is preparing for your interview or exams like CSIR NET, GATE or BARC, I would say you are at the right place. So in today's video, we will be learning about one of the most fundamental concepts in life science, which is immunology. Considering the amount of research or the whatever the situation which is going on around us, the diseases, the spread of the diseases, immunology is an important thing in life science at this moment. And today, particularly, we will be studying about innate immunity and how the physical barriers within this innate immunity portion act as body's first line of defense. Okay, so let's get started. So before going into the different mechanisms that how it is supporting or acting as a first line of defense. Let's first know about some basic terminologies or essential terms, I would say, in immunology, because if you sit for any interviews as well, uh, they are going to ask you about these basic terms. It's some basic questions, but it's going to be there in your interview. It's going to be there in your papers as well. So what are these terms? The terms include the immunity, immune system, and pathogens, antigens, and antibody. So I'm quite sure that with antigen and antibody, most of you are familiar with these two terms, but still, let's just start from the beginning. So immunity. Immunity simply means a body's defense mechanism against any kind of potentially harmful material or foreign agents. Right? Immune system means a complex network of cells, tissues, and organs which are protecting us from the pathogens, or a complex network of cells, tissues, and organs that help us to perform this immunity phenomenon. So I have been asked this question a lot. That does the immune system helps in performing immunity? So the answer to this question is yes. Yes, again, it's a very basic question, but this type of questions are asked. So you can uh, be very sure with that. Now the next term is pathogens. Now, any organism that can cause any kind of a disease in our body or any kind of a disease causing agent is considered as a pathogen. And if we consider human pathogens, then they are grouped into four different types. One is bacteria, viruses, fungi, and parasites. Now, under this, there could be different kinds of subfamilies and subclasses which are present, which we will be learning in a different video. But pathogens, like if you are asked that what are the different classes of human pathogens, it's mainly of four types, which is bacteria, viruses, fungi, and parasites. And the most common or most renownedly used two terms, one is antigen and another one is antibody. So an antigen is any kind of a substance or foreign substance that triggers an immune response in our body, and antibody is a wide-shaped glycoprotein or a protein which is produced by the B cells that binds it specifically to the antigen and tries to, eliminate them. So now we know that there are two types of immunity. Okay, two main types of immunity which we have in our system, our post defense mechanism. If we consider, there are two types. One is innate immunity, another one is the adaptive immunity. Innate immunity is a non-specific and immediate response of creates an immediate response, and it is present from birth, in contrast of adaptive immunity, which is highly specific and develops over time and has memory. So let's now just zoom into innate immunity portion. Okay, so innate immunity, as I said, that it's our first line of defense against any kind of a foreign particle or a foreign material or any kind of a pathogen. Now it's like you can think of it as a security system which is always active. It's never sleeping, always active. And it's also called a natural or non-specific immunity. Why it is known as non-specific immunity is because whatever the foreign material you give, whatever the pathogen it is, it will act against it in a similar manner. It's not like for pathogen X, it's going to be overstimulated, and for pathogen Y, it's going to be understimulated. There is no such differentiation in case of innate immunity. In innate immunity, for like every pathogen, if there are 100 pathogens, for those all the 100 pathogens, the immunity is going to function in the same way, and the function is going to be, and like the response which is elicited by this immunity or this immune system is going to be the same. So that is why they are considered non-specific immunity. Immunity is also known as natural immunity or inborn immunity. Why? Because it's a present from the birth itself. So we are born with it. Whatever which is present in our DNA, it's going to be our own thing. Whatever other materials which is going inside our body, coming inside our body, or entering in our body, they are going to be considered as a foreign material or a foreign particle. And for such a things, different like our immune body is competent enough to function against it, so that that particular foreign XYZ material could be killed or could be destroyed, okay, or could be solubilized. Now, in case of innate immunity, it's an inborn immunity. It's a present as a part of an animal's innate structure or inherited as a part of animal innate structure. So whatever you are inheriting from your parent, from your parents, whatever, however the structures are, whatever the genes which are present, there, whatever is there in your DNA, those things are going to be your own, and anything apart from that is going to be a foreign particle or considered as a foreign particle. And what does it mean from animals innate structure is that the different components here from animals innate structure, I want to specify the different components which are present in our innate system. For example, the physical barriers which we have, the chemical barriers, improve with repeated exposure, and no memory response is generated in innate immunity. So, like, there are some specific examples or exceptions there, but in general, there is no memory response which is generated, and it does not improve with repeated exposure. So that is the line which differentiates between or distinguishes innate immunity from that of the adaptive immunity. So now let's look at the components of innate immune system. So there are different components. One is the physical barriers, like skin is there, mucous membrane is there. Okay. Second is the chemical mediators, which includes enzymes like lysozymes, acids, etc. For cellular defenses, there are cells which are present to create this innate immunity. One of the prominent example is natural killer cells or the NK cells, or also there are macrophages or phagocytes which are present. And another one is the inflammatory barriers, which helps to, you know, if any infection is taking place, so whatever we learn about a hypersensitivity or allergic reactions, so or the interleukins, the cytokines which are involved in performing the chemokinesis of the of the macrophages in order to phagocytose a particular cell, or the natural killer cell to perform an ADCC, like antibody dependent cellular cytotoxicity, those functions. So inflammatory barriers includes all those cytokines and interleukins which play an important role. So they do fall under innate immune system, not completely under adaptive immune system. So it act as a, what should I say, a bridge between an innate and adaptive immune system. So today we will be mostly focusing about the physical barriers. Now there are different types of physical barriers. Mostly these are the important one. Apart from this, questions are not going to come. I can give you the guarantee. Now, what are the different types we will be studying today? Skin, mucous membrane, respiratory system, gastrointestinal tract, and genitourinary tract. So let's start with skin. So you know about skin. The basic one, we know about it, that it acts as a mechanical barrier to any kind of a microbial invasion. It acts as a protective shield. The outer layer is made up of keratinocytes, which are thick cells, and they are closely packed cells. And these keratinocytes are the cells which produces keratin, which is a kind of an intermediate filament, which makes the surface tough and water resistant. Okay, so let me know about skin, that there is a constant shedding of this keratinocytes, or the skin cells which is taking place. Uh, that is why, like, out of context thing, I'm saying that girls, maybe they can, you know, more accurately recognize what I'm saying is we do scrubbing and all those things, right? So to remove the dead cells. So what are these dead cells? They are the shedded cells, or the shedded keratinocytes. So why this continuous shedding is important, of continuous shedding of the skin cell is important, because some of the microbes may adhere to the surface of the skin cells. So once they are adhering to a particular cell, okay, but that particular cell is a shedding off, as a result of which the microbe that has adhered to that cell, it will also be shredded off from the body. So that is why constant shedding of the skin, a continuous shedding of the skin, removes the microbes that adhere to the surface of that particular cell, and it again helps in preventing the any kind of microbial invasion in our body. Also, skin has a slightly acidic pH, which ranges between 5 to 6, and this acidity is due to the sebum, or the oil, which is produced by the sebaceous gland, along with sweat glands and the organic acids. These organic acids are mainly produced by the commensal bacteria, so like Staphylococci. Commensal bacteria means the bacteria which is usually residing in our skin. Okay, so even our high salt concentration and periodic drying also make a hostile environment for the microbes to invade the skin and penetrate through it, and cause the pathogenesis, or cause the infection. So the most important physical barrier, or one of the important physical barrier, is the skin, and the reasons I have just stated here. Now the next one is a mucous membrane. Fine. Now the location, if we see, the mucous membrane are present in the areas like the eyes, respiratory tract, GI tract, and urogenital tract. They are made up of stratified epithelium as well as mucus. So what makes them so special? They act both. Okay, this is a type of a class which act as both physical as well as chemical barriers. Why I'm saying it? Because the secretion of the mucous membrane do contain chemicals such as lysozyme, which breaks down the bacterial cell wall. Lysozyme specifically cleaves the bond that joins the NAM and NAG together to form the bacterial cell wall, especially in the gram-positive bacteria. Because we know that in case of, like we have read about gram staining, we know that in gram-positive bacteria there is a thick peptidoglycan cell wall which is present. And this thick peptidoglycan cell wall, once this thickness is gone, the gram-positive bacteria is not going to survive. So, and the bond which is present between these alternative units of NAM and NAG, the bonds in between them is cleaved by this lysozyme enzyme, which is present in the secretion of the mucous membrane. So it is acting as a chemical barrier. Another example of what is present in the secretion is lactoferrin. Lactoferrin is an what should I say? It binds to free iron, and as a result of which, in the blood plasma, if any kind of pathogen is surviving in the blood, or in the blood plasma, or in the blood, why it is surviving there? Because it got the environment which it needs to survive. Now, if you take the iron from there, so lactoferrin is present, it will bind to the free iron, as a result of which the iron is trapped now. Iron acts as a macronutrient in case of a pathogens to survive. So the free iron content in the blood plasma will be low, as a result of which it will deprive the microbes from the essential nutrients. As a result of which, the microbe is not going to survive. The microbe is eventually going to be killed. And this lactoferrin is mainly released by macrophages and PMNs. PMNs, the full form of it is polymorphonuclear cells. It is another name for neutrophils. You can note it down. Now, the second point is the secretory immunoglobulin A. Why I'm saying secretory immunoglobulin A is because if you electrophores this secretory immunoglobulin A, there are going to be four chains that you are going to see. One is the heavy chain, another one is the light chain, another one is the J chain. Because in case of secretory IgA, it's going to be a dimeric immunoglobulin. So once you are doing a native, once you are going to do a SDS page or a reducing SDS page, what you will get are four bands. One is the heavy band, one is the light band. Fine. Another one is going to be the J chain, which is going to join these two dimers together. Another one is going to be the secretory protein, the secretory component or the soluble protein. So this soluble protein, what it does, it prevents the proteolytic sites in the immunoglobulin molecule or the immunoglobulin A molecule, as a result of which any kind of proteolytic enzyme which could be present in the mucous membrane is not going to cleave this dimer. And this once this immunoglobulin is present, it blocks the microbial adherence to any kind of an epithelial surface. And it is the most abundant immunoglobulin in the mucosal secretion. Why? Because of the presence of the soluble protein or the secretory component. Another one is lactoperoxidase, which produces reactive oxygen species like superoxide radicals. And we all know reactive oxygen species or superoxide radicals, they are too toxic for any kind of a microorganism. And additional, if you say defense actions, where mucous membrane plays a very important role is your tears. Your tears also helps to wash away the pathogens. If any kind of a, pathogen is entering, or any kind of a small fly, very small fly is entering in your eye, what do? You start crying, or some water comes out of your eye. Why such situation happens? Because it tries to remove whatever the foreign material has gone inside your eye. So that's why. So now we have studied about two main components. One is skin, another one is the mucous membrane. So let's skip to the next part, which is respiratory tract. In respiratory tract, it is a sophisticated cleaning system, I would say, because mucosal lining is present here in respiratory tract as well. I have mentioned it in the previous slide as well, because mucosal membranes are present in the respiratory tract, in the GI tract, in the lungs. It is also present in the nasal cavity. It is present in the eyes, etc. So mucosal lining is present. So this mucosal lining helps in trapping the microbes on the moist surfaces. Cilia and hairs are present, which helps in the nasal cavity, as I just mentioned, to trap the particles which are greater than 10 micrometer in size. The mucociliary is exclarator and the blanket. What this do is they help in constant movement. So they are always beating. So the cilia always beats constantly. It is beating towards the pharynx, as a result of which it pushes out the mucus. In this mucus, if any kind of microbes are trapped, so they are also pushed out, and it traps very smaller particles that reach to lower airways. Because usually here I have what I have said, that cilia and hairs, they are going to trap particles of greater than 10 micrometer. What about the smaller particles? If it is reaching the lower airways, then this mechanism, the mucociliary escalator as well as the blanket, it helps, like it consists of a numerous number of cilia which will constantly be towards the pharynx to push the mucus and the trapped microorganisms or the microbes within the mucus out of the system. This respiratory tract also helps in humidification. Humidification, as a result of humidification, what happens is that if a microbe is present in the airway, that microbe will swell, as a result of which once the microbe is swelling, it is reaching the lower airways because it is smaller in size. Now, macrophages, one thing we should remember, that macrophages, if it's a smaller substance, macrophage is not going to recognize it, and it's not going to phagocytose it. So for humidification, the microbes swells. Once the microbe swells, it increases in size, as a result of which the macrophages will be able to recognize it, as a result of which the phagocytosis will take place. Okay. The next is reflexes. We see that if, for example, anything goes inside our mouth, we start coughing, we start sneezing to expel the pathogen forcefully out from our airways. Salivation also helps in flushing the microbes from nasopharynx into the stomach. Okay, and like deep in the lungs, alveolar macrophages are present, which act as the final guardians, I would say, ingesting and destroying whatever the inhaled microbes, if it has gone. So they are also again killed by the alveolar macrophages, which is present in our lungs, because in the residing tissues and different residing tissues, the macrophages are given different names. So yeah, that is the another point. Now the next will be gastrointestinal tract. Now the GI tract is a hostile place for microbes. Why? Because it is acidic in nature. It is acidic because of the presence of the gastric juice, which contains hydrochloric acid, different proteolytic enzymes, as well as the mucus. Inside the mucus, as I have already said, there are secretions of lysozyme, lactoferrin enzyme is there, different peroxidase enzyme is there, different enzymes are there. So ultimately, it kills most of the pathogens. Now, the intestinal secretion also contains bile, pancreatic enzyme, brush border enzyme. Now, here I have included GALT or gut associated lymphoid tissue for immune surveillance because it's there in the newest edition of QB. But this gut associated lymphoid tissue, as the name suggests, it's not under the completely not under the innate immune system. Okay, it acts as a bridge between the innate towards the adaptive immune system, because here in this lymphoid tissues, gut associated lymphoid tissues, there is some residing B and T cells which are present, which can again recognize a particular pathogen and secrete different types of antibodies. Mostly or mainly, it's going to be the secretory immunoglobulin A that I have talked about previously. But yeah, there are residing B and T cells. So it's not completely under innate immunity, even though I have written it here, but it acts as a bridge between the two. Now, gut flora or the gut microbiome, it plays a very important role to compete with the pathogen, because in gut microbiome, we know that there are different types of different population of the pathogens which are present, right? So it consists of opportunistic pathogens as well. So we have good bacteria, we have bad bacteria. If the amount of good bacteria depletes, then the opportunistic pathogens or the bad bacteria gets area or other nutrients are necessary for them to grow, and as a result of which the pathogenesis or the disease could take place. So again, gut microbiome do plays a very important role in competing with any kind of pathogens. Another important cell in intestine, I would say, smaller intestine specifically, is the Paneth cells. Okay, so it releases again enzymes such as lysozymes as well as cryptidins. Cryptidins is another name for defensive. We know that in case of T cells or in case of natural killer cells or NK cells, what they perform is they produce gran enzyme and perforins. Right? What perforins do is they create a channel so that the gran enzyme could enter and thereby cleave the cell or destroy the cell. Peptidins again act as the pore forming or performin or the defensive enzyme. So it produces holes, different types of channels or different types of pores in the plasma membrane of the bacteria or of the microorganism, as a result of which an osmotic shock could take place, as a result of which the bacteria or the microorganism is going to die. Okay, so it creates a membrane pore and cell lysis will take place. Now, the most, what should I say, unique one is the genitourinary tract. Genitourinary tract has its own set of defenses. Okay, so estrogen is there. It's a hormone regulated defense system is also included. Then sterility or mechanical defense is also included. Chemical barriers are also included in this tract. So let's just go into the hormone regulated defense first. So here estrogen stimulates glycogen secretion in the vaginal lining. So once this glycogen is produced, we have like I know many people will know about this, that in vagina we have a huge amount of huge population of a bacteria, specific bacteria, which is lactobacillus acidophilus. You can see that it's like the amount, the microbial load, if I consider, is 10 to the power 8 lactobacilli per ml. So once the estrogen stimulates the epithelial cell to produce glycogen, the lactobacillus acidophilus will act on glycogen, so that breakdown of glycogen could take place to form lactic acid. Once lactic acid is produced, it creates a very acidic environment for the vaginal secretion. as a result of which it inhibits most of the pathogens, because most of the pathogens are not going to survive in that acidic environment. The cervical mucus also contribute to defense via the antibacterial activity that it has. Now, the urine itself has a flushing action. It's like, if we consider sterility, kidney, ureters, urinary bladder, they are normally sterile. And urine is also sterile within the bladder under healthy conditions. And few bacteria may be present in distal urethra. Why this could be present is one of the main reason for this like presence of the few bacteria in the distal urethra is onset of a UTI or urinary tract infection. And this question was asked in BRC, like in the examination, why UTI are more common in case of female urethra than that of the male urethra. Why it is common? Because it's based on the anatomical difference that we had. Where in female urethra, it's a 5 centimeter long. So it's a shorter than that of the male urethra, which is 20 centimeter long. So of course, it's quite common for the bacteria to enter a shorter distance, to travel a shorter distance and cause the disease. So UTIs are 14x more common, 14 times more common in females due to the shorter urethra and anatomical proximity to microbial entry points. But still, since we have different mechanisms to prevent those infections, we don't so commonly get it, but due to whatever the if hygiene is not maintained and if things like that, you may get UTI, and for females, it's more common because of the anatomical difference that we have. So that's it, I guess. So that's it. That's a wrap up to the physical barriers of innate immunity. Now, these defenses are always active. Okay, keep in mind, always active, preventing countless infections without us even realizing it. So, in the next video, we will dive into the different cellular components of the innate immunity and the chemical mediators. So, at the end, if you found this video helpful, give the video a like, subscribe, and comment below with any specific topics you want me to cover, and whether it's from the exam perspective, it's from for the IISc PhD preparation, any PhD preparation, any interview preparation, I'm here to help you. So see you in the next one. Bye bye.